Thank y'all for coming. I'm going to be talking about my research on the immunological response differences between worker and drone honeybees. So, as we all know, honeybees are important. They offer us honey, and then they're also and they also offer us pollination services. So on the x-axis, you can see the year, and then on the y-axis, you can see the gross revenue for each source. And then the blue bars represent the honey cells, and the orange bars represent income from pollination services, and then the gray represents other apiary product sales. And so I like to point out that there's the honey and the pollination services make up the majority of the revenue for honeybee services, and so it's well over $300 million. And so honeybees are important. They offer us, they're our primary pollinators, and then they pollinate up to 60% of plants globally. And so in this image here, you can see honeybee colonies on like a semi-truck. And so what's going on here is that apiarists in the north will rent out their honeybee colonies to farmers in the south during the winter. And those farmers are able to increase their crop yields by having extra like pollination by the honeybees. And so that just shows another like service that honeybees are offering us. And so, as I had mentioned, honeybees are primary pollinators, and so there are certain crops that are only pollinated by honeybees. And so these are a couple of examples. You have almonds, apples, berries, melons, and squash. And at like face value, you may not think, oh, I don't eat that many almonds per year, per month. But almonds have other products that they are used to make, such as like almond butter, almond oil, almond flour, almond milk. And so all of those are like major components of the economy and like economic like sectors, and even in like the agricultural sector. And so just to highlight that honeybees, that they do offer us services aside from honey and pollination services. And pollination services do make up a large majority of the annual revenue generated by honeybees. And so, as we know, they are essential for agriculture. And then the human population is increasing. And so as the human population increases, we need to be able to sustain our food source. And then, however, like crops are flatlining or they're decreasing, which is not the best things considering that the human population is increasing. And then honeybees do make up an estimated about $50 billion annually for just the United States alone. And that just signifies the importance of honeybees. And so that just emphasizes the importance that honeybees have for certain sectors within the economy. Okay, cool. However, there's a problem. And the problem is that honeybees populations are declining. And so in 2006, there was a phenomenon called colony collapse disorder that began to like come to light. And this is essentially a phenomenon where a majority of the worker honeybees are leaving the honeybee colony without any indication. And so this is essentially happening overnight, and it makes the colony unstable and like unable to function. And so this is causing bee populations to decrease over time. So if we look at the image over here, you can see on the x-axis the years, and then starting in 2007, since that's whenever data first started being collected, and then you have the percent loss on the y-axis. And so this is important to look at when it comes to overwintering, which was where apiarists rented out their honeybee colonies to the farmers in the south, because in the year 2023 to 24, about 59% of honeybee colonies were lost in that overwintering phenomenon. And that's not good because apiarists in the north are sending their bees down here, and 59% of those are dying, and then we don't really have anything else to send back. We're just a small majority. So it makes it harder to increase crop yields in the north. And then as you can see that there's like this upward trend, and so we want to focus on the red image, on the red graph part of the graph. That's the total percent of honeybee losses. And so while it is fluctuating, there is an upward trend in the percent loss of honeybees. And so while we don't know what exactly is causing colony collapse disorder, there are various potential factors, such as climate change and pesticides. So an example of climate change is in Georgia, they're experiencing extreme freezes one week, warmer temperatures the next week, and then another cold snap in the following week. And so that's just really hard on the colony to be able to maintain itself. And so that could be a potential factor contributing to colony collapse disorder. And then also pesticides, like the neonicotinoid. That one is popular in like the agricultural industry for pests, and it can have harmful like side effects and unintended side effects on honeybees. And so that just makes it harder for those colonies to be able to maintain themselves. And so we think that there are immunological factors contributing to colony collapse disorder, that their honeybees are having like essentially this ability to withstand these factors, but they're only able to do so as strong as their immune system is. And so a few ones that I point out are insecticide exposure, viruses, pathogenic bacteria, fungi, and then parasites. Those are some of the ones that we are focusing on. And so we think that there's a synergistic effect occurring. And so that means like within any one of these given factors, they're able to like tolerate it, but once each factor crosses a certain threshold, they're not really able to sustain themselves. And so that's what's contributing to colony collapse disorder. And so just a look into the colony, there is a hierarchical system. So there's workers, queens, and drones. And so within each caste of this hierarchy system, there are differing roles. And so the worker honeybee, they are responsible for maintaining the colony. So they make up the majority, they're the largest caste in the colony. And so they maintain the colony, and they're also scavenging, they're looking for food and all those other things. And then there's the queen, who's solely responsible for reproduction. And then there are the drones. And the drones essentially do nothing. And so the queens and the workers, those are the female honeybees, and then the drone is the male. And so the drone essentially just like sticks and hangs around in the colony, and they're just leeching off of resources. And then once they reach sexual maturity, they fly off and mate with the queen of another colony. So they're not really contributing to the colony, they're just like leeching off of the resources. And so we propose that there's potentially differing energy allocations for immune responses given these different duties with each caste. Yeah. And so our research question was, are there immunological differences between worker and drone honeybees? And so over here you can see the drone is labeled B, and then the worker is A. You can tell by the size of the abdomen and or the thorax, and then also the size of the eyes, how close they are. The drone has eyes that are closer to one another. And so to conduct this experiment, we started off by honeybee collection. We have colonies here on campus, and they're up by the garden. And so we'd use a little centrifugal tube with a hole drilled into the cap. We'd go out there and just collect the honeybees, and then we would bring them back to the lab and incubate them at 26 degrees Celsius, kind of mimicking that same temperature of the colony. And then we divided them into different experimental groups. So I already talked about collection and incubation, and so now we go into like how we divided them into experimental groups. So the basal immunity is one group that just gave us like a baseline of what's going on immunologically with honeybees. So you can think of this as like the baseline, the norm of what the honeybee is supposed to be eliciting, and then we divided it into an activated immunity. So this group, as it sounds, had an activated immune response. And so this immune response was activated using an LPS solution. So LPS solution is a lipopolysaccharide, and this is like a macromolecule chain that's on like bacteria on the outer wall. And so this is easily bought. So essentially that LPS is like cleaved off of the bacteria, and then we can administer it to the honeybee, and the immune system believes that there's an immune response going on, or there's a pathogenic threat, and so the honeybee is able to elicit an immune response. And so that was what we intended. And then depending on the group it was in, we allowed time to elapse for 0, 1, 2, 3, or 4 hours. just to get an idea of like a time series. And then after that, both groups were, we processed their hemolymph. And so hemolymph is essentially the like bee's blood, and so we had to extract that from the honeybee. And so we started off by dissecting the thorax, which is that middle part between the head and the like the tail. And so once we dissected that, it removed the wings and legs, and then we used a mortar and pestle in a little microtube and grinded the thorax using like a mortar and pestle. And then we homogenized those samples, so we use a machine to kind of like lyse up those cells, anything that we missed using the mortar and pestle, just to make sure we get all those proteins out of the cell. And then we stored it at minus 80 degrees Celsius, and because that's just like the temperature that things stopped altering. And then we ran some colorimetric assays. So the first assay we ran was a Red 660 assay. We ran this assay because it allows us to, first off, we use a different method, and so we weren't standardizing the amount of hemolymph we extracted from the honeybee, and so we needed to get an idea of the total concentration of proteins, that way we can normalize the data. And so that's what that one was for. And then we did a prophenol oxidase assay, and a prophenol oxidase assay prophenol oxidase is a precursor molecule in the melanin synthesis cascade, and so honeybees kind of have like 3 different ways it can elicit immune response. And they can use antimicrobial peptides, they can use encapsulation, and they can use phagocytosis. So we focus on the encapsulation one, and so we use prophenol oxidase as a proxy for immune activity in the melanin synthesis cascade, and that's what we were measuring. And then just here you can see like the color differences in those 2 wells, just that there's activity going on there. And so these are our results. So on the x-axis, we have time, and then the basal activity. So the basal activity, again, is just that baseline of what's going on. And then we have time in 0, 1, 2, 3 hours. And then on the y-axis, we have prophenol oxidase activity. That just is a proxy that we're using for immune activity. And then we have the white bars are representing the workers, and then the gray bars represent our drones, so the males. And then I just want to point out that time cast and then time and cast were statistically significant in this, so each one was important. So first off, I'll point out this bar. This is the highest one, so this just means that that 1 hour, the worker bees were eliciting a strong immune response. There was high immune activity. And then if we would look at the drones, it seems to be the same all the way around. There are fluctuations, but it seems to be like baseline similar activity. And same thing kind of for the workers, but except at that 1 hour mark, there's that spiked immune response. And so from this, we were able to conclude that there was a significantly higher PPO activity between cast and time. And then this has the effect to, the possibility to affect immunological responses. And so the workers, they're out and about and they're scavenging. So they're constantly exposed to various pathogens, and so we think that the honeybees are kind of like spiking their immune response because they don't want to bring that potential pathogen back to the colony, and so they just ramp up their immune response, and they're able to kind of combat that pathogen before they return to the colony. And then drones, since they don't really leave the colony, they didn't really like evolve this like immune response, because once they leave the colony, they mate and then they die, so there's not really a need for that. And so going forward, we want to run some antioxidant assays to kind of get everything going, and then, or get a better idea of what's going on, and then we want to narrow in on that time sequence. So there's another group in the lab doing that. They've kind of narrowed it in from 1 hour to different time intervals, so we can kind of get a more defined bell curve of what's going on. And so we just want to say thank you to St. Edward's and the Bromard Research Fund for allowing us to do this. Thank you.
I have 2 questions. One, if you were to test this on, I know you don't have too many queen bees, but do you think the queen bee would have a similar immune response to the worker or the other one? And then two, did you get stung while collecting?
So for your first question, I honestly am not sure what the immune response would be like, but since queen bees are kind of like start off as like worker bees, I would assume they have a similar immune response to the workers, and then they also receive different nutrients as they are like metamorphosized into a queen bee. But I mean, I don't know for sure. I don't have a definitive answer. And then no, I did not get stung.
I have a related question. I think it's the same question, but articulated from a different standpoint, which is, do you think the difference in immune response is more an inherent characteristic of the workers versus the drones, or would it be acquired as a result of the workers going out into the world, which the drones really don't? And is there a way that you can test that? And the queens help answer that question.
So that's a great question. I'm not sure about what we're able to determine using the queens, since there's just such a limited majority of them. But since honeybees do have an innate immune response and not an adaptive immune response, I don't believe it would be like an acquired. So there are, I was talking about this yesterday with Dr. Stevenson, but there are ways that the innate immune response kind of is primed to tackle those various like pathogens that it comes into contact with, but I'm not sure on the exact like mechanism of which that would have evolved.
Kind of along similar lines, do you think solitary bee species would not show that because they're not bringing something back to a hive?
What do you mean by solitary bee species?
Well, non-social bee species.
Yeah.
Not all bees live in a colony with friends.
Mm-hmm. I'm not sure on what exactly that would be.
I know you don't know, but what do you think? Because your hypothesis is that this evolved so that you protect the hive.
Mm-hmm.
There's no hive to protect, I think.
Mm-hmm. That's a good
And
question.
would that be a way of testing that hypothesis?
I think that's a good question, and something that would be interesting to look into, seeing how the immune response is correlated with like social behaviors of the honeybee. I would think that there would be a higher immune response since they're just being exposed to those various pathogens, and they can like kind of prime themselves to be like come in contact with those. But I'm not sure on how that would.
All right, thank you, Jared.
Now, I'd like to invite Jared's mentor, Dr. Matt Stephenson, to present him with his stole and cords that he'll wear during graduation as an honors student, and to say a few words about Jared.
I was told I have to come up with a few really nice things to say, so I spent a talk thinking. I had to search. But Gerad has been working in my lab for about 3 years. He has been doing a fantastic job. Has also continued to run assays on projects that I currently have running in my lab, and has always been a really good team player and always willing to come and pick my brain about bees. So congratulations, Gerad. Yeah.